Good morning. It's June 17th, and this is your daily brief in science. Here's everything you need to know. Diving into the depths of our oceans, researchers have discovered compounds with the power to combat some of today's most challenging medical issues. A study published in Marine Drugs unveils a treasure trove of potential from the marine-derived fungus Talaromyces variabilis, revealing 3 new compounds, 8 to 9 EPI crodromonins, which show promise for gastric cancer therapies. This discovery opens a new frontier in the search for cancer treatments, highlighting the untapped potential of our oceans. Further exploration into the marine world has led to the discovery of new cyclic pentapeptides, avellanins DG, in a mangrove-derived fungus. Notably, Compound 2 has shown insecticidal effects against Culex quinquefasciatus larvae, pointing towards innovative solutions for pest control. Additionally, a collection of 9 compounds from marine sources and Aspergillus fumigatus giximt 03099 have been identified with insecticidal and antibacterial properties, underscoring their potential in developing new antimicrobial strategies. In another significant find, the marine-derived fungus Penicillium rubens BTBU 2021-035 produced 4 new compounds, including penirubenones A and B and penirubenomides A and B, alongside 9 known compounds. These substances exhibited notable antibacterial and antifungal activities, with 1 showing effectiveness against Staphylococcus aureus and 2 demonstrating synergistic effects with rapamycin against Candida albicans. This points to the critical role marine natural products could play in developing novel antifungal drugs, especially crucial in combating drug-resistant strains. These findings not only highlight the ocean's vast potential as a source for groundbreaking medical treatments, but also address the urgent need for new antifungal therapies in the face of increasing drug resistance. The exploration of marine-derived compounds is proving to be a promising avenue in the quest for innovative therapeutic solutions across various medical fields. In a breakthrough that could redefine our understanding of the quantum world, physicists at CERN, led by Regina Demina of the University of Rochester, have made a monumental discovery involving quantum entanglement and top quarks, the heaviest known fundamental particles. This finding not only challenges existing paradigms, but also opens up thrilling new prospects for the future of quantum research. The team observed quantum entanglement between unstable top quarks and their antimatter counterparts at distances that surpass the speed of light. This observation is significant because it demonstrates the persistence of entanglement, an elusive phenomenon that has puzzled scientists for decades in systems involving the heaviest of particles. Such entanglement at great distances provides a new lens through which the quantum realm can be explored. The implications of this discovery are vast, potentially paving the way for new physics beyond the Standard Model, which is the current best theory describing the fundamental particles and their interactions. Understanding how top quarks entangle could offer insights into the evolution of the universe and the forces that govern it. Moreover, this research illuminates the potential of entanglement in quantum information science fields, such as cryptography and quantum computing, suggesting new methods for secure communication and advanced computational capabilities. This collaborative effort at CERN marks a significant milestone in the quest to unravel the mysteries of the quantum world. It highlights the importance of international cooperation and the relentless pursuit of knowledge in pushing the boundaries of what we know about our reality. The findings from this study not only contribute to our comprehension of the quantum universe, but also hint at the untapped potential that quantum entanglement holds for future technological advancements. As the world grapples with the stark realities of climate change, Antarctica and South America have become the latest epicenters of extreme weather events, painting a vivid picture of our planet's shifting climate dynamics. In late winter 2023, Antarctica was gripped by extreme cold spells, setting record-low temperatures, while South America experienced the opposite extreme with record-breaking high temperatures. This stark contrast underscores the unpredictable nature of our changing climate. Researchers have pinpointed 4 distinct cold phases during this period in Antarctica, with strong negative anomalies in the mid-tropospheric atmospheric environment, identified as a contributing factor to the extreme cold. Concurrently, Antarctica witnessed a record-breaking minimum sea ice extent in 2023, a phenomenon likely driven by climate change. The rarity of such an event is highlighted by the estimation that this level of sea ice loss would typically occur once every 2,000 years in the absence of climate change. The findings, published in geophysical research letters, were derived from data collected by automatic weather stations, offering a detailed analysis of these unprecedented weather events and the sea ice extent. This research not only sheds light on the immediate effects of these anomalies, but also emphasizes the need for enhanced preparedness for future logistical challenges in Antarctica. The long-term implications of sustained low sea ice levels are a growing concern among scientists. They warn of potential lasting impacts on Antarctic ecosystems and weather patterns, signaling a critical need for continued monitoring and research to understand and mitigate these changes. In the quest to revolutionize the care for traumatic brain injury, or TBI, patients, researchers are zeroing in on aquaporins, or AQPs, as potential game changers. These proteins, crucial for water transport in the brain, are now being studied for their roles as biomarkers in TB1, offering a glimmer of hope for improved diagnostics and patient care. A recent study has shed light on the significant role of AQP2 in patients with acute and chronic subdural hematoma. The findings reveal that AQP2 levels correlate with the volume of chronic hematoma and midline shift, marking a promising step forward in understanding chronic TBI cases. This discovery is particularly intriguing as other AQPs did not show significant correlations in acute subdural hematoma, highlighting AQP2's unique potential in signaling inflammation and disease severity. The investigation into AQPs extends beyond AQP2, encompassing AQP4, AQP9, AQP1, and AQP2, each believed to play a distinct role in the pathophysiology of TBI and intracerebral hemorrhage. AQP4 is associated with brain edema. AQP9 could assist in water removal. AQP1 is linked to edema formation, and AQP2's involvement might influence astrocyte activation and clinical outcomes. Understanding how these proteins function and interact in the context of TBI is crucial. It opens the door to developing targeted therapeutic strategies aimed at managing brain edema and enhancing patient recovery. The research into AQPs as biomarkers for TBI represents a pivotal step towards achieving this goal, promising to refine diagnostic tools and therapeutic approaches for those affected by such devastating injuries. In the face of growing environmental challenges, a groundbreaking study sheds light on innovative strategies to bolster maize resilience against drought and other abiotic stresses. This research, published in the Plants Journal, delves into the genetic and microbial realms to unlock new avenues for agricultural sustainability. The study centers on the ZMATG3 gene in maize, exploring its pivotal role in the plant's response to various abiotic stresses, including drought. Scientists discovered that overexpressing this gene significantly enhances maize's tolerance to adverse conditions such as high salt levels, mannitol-induced stress, and low nitrogen availability. But the research didn't stop at genetic manipulation. The team also investigated the use of plant growth-promoting bacteria, PGPB, as a means to further improve drought tolerance in maize crops. Specifically, strains of Azospirillum brasiliense and Bacillus speas were tested, showing promising results in boosting growth parameters of maize under moderate drought conditions. This dual approach, combining the overexpression of the ZMATG3 gene with the application of beneficial microorganisms, highlights a holistic strategy to enhance plant resilience. By tapping into the genetic makeup of crops and leveraging the symbiotic relationships with microorganisms, this research offers potential solutions for improving agricultural productivity in the face of challenging environmental conditions. The implications of these findings are far-reaching, suggesting that future agricultural practices could significantly benefit from integrating genetic and microbial technologies. This could pave the way for more robust crops capable of withstanding the increasingly unpredictable climate, ultimately contributing to global food security. This has been your daily brief in science. To read more about these stories, follow the links in the episode bio. You can also subscribe to these updates via email at www.brief.news. For more daily podcasts about the topics you love, visit brief.news/podcasts. Tune in tomorrow. We'll be back with everything you need to know.